Sea urchin development. Fertilization. The animal pole and the vegetal pole. Here there is unequal equilateral cleavage to produce two cells. These two cells then merge into unequal equatorial cleavage to produce four cells. These four cells show the ectoderm, endoderm, and mesoderm. The 8 cells represent the blastomeres. On the animal tier, their meridional division of the 4 animal tier cells result in 8 mesomeres. The vegetal tier, unequally equatorial cleavage to produce 4 macromeres and 4 micromeres. Here's the 16-cell stage where the mesomeres divide equilaterally into 2 tiers, AN1 and AN2. Present also are the macromeres in yellow and the micromeres in red. Here's the 32-cell stage where the macromeres divide marginally upwards into 8 cells. The 64-cell stage shows the animal pole with AN1 and AN2, and the vegetal pole with VEG1 and VEG2. Also, at the bottom, you see micromeres divided unequally, forming 4 smaller micromeres. Here's the blastula. After micromeres have slowed cell division, all cells are the same size and now have a blastocell layer on the inside and a hyaline layer on the outside. Invagination begins and the primary mesenchyme moves towards the center. Primary mesenchyme was originally the mesoderm. Gastrula. Now invagination of the endoderm occurs. Secondary mesenchyme occurs at later gastrulation. Here you see the ciliary tuft, the secondary mesenchyme in yellow, the endoderm, ectoderm, primary mesenchyme which was once the mesoderm, and the gut. The primary mesenchyme becomes a skeletal rod, the mouth forms an oral hood, and the anus becomes a small centered hole. Here you see the Pluteus larva. This is the final gastrulation stage in sea urchins, and it is primarily bilateral. This Pluteus larva then undergoes metamorphosis to form the sea urchin. Here is the beautiful sea urchin.